An architect sits at a drafting table. Blueprints spread out, rulers and compasses at hand. Every line on the blueprint is a vector. Every transformation, rotate, scale, mirror is a matrix operation. And hidden inside every structure are natural frequencies that determine how it responds to stress. This is linear algebra. And every algorithm in computational biology, PISA, UMAP, and network analysis, even neural networks, speaks this language. A vector is an arrow, direction plus magnitude. In biology, a single cell's gene expression profile is a vector in 20,000 dimensional space. Each dimension is one gene. The vector's position tells you which genes are on and how strongly. Plot 2,000 cells, and each is a point in this space. Cells with similar expression clustered together. Cells with different expression are far apart. All of genomics operates in this vector space. A matrix is a grid of numbers that transforms vectors. Multiply a matrix by a vector and you get a new vector, rotate, stretch or sheared. The data matrix itself is a matrix, rows are cells, columns are genes. Each entry is the expression of one gene in one cell. Multiplying this matrix by a gene weight vector gives a score for each cell, a weighted combination. This is what PCA, regression, and neural networks all do. Multiply two matrices, and you combine their transformations. Rotate, then scale in one step. But order matters. A times B is not B times at the covariance matrix. X transpose times X captures pairwise relationships between all genes simultaneously. Each entry is the covariance between two genes. This single matrix contains everything pika and gana need to find structure in the data. The dot product measures alignment. How much does vector A point in the same direction as vector B? If they're perpendicular, the dot product is 0, no relationship. If parallel, it's maximal, perfect correlation. Correlation is a normalized dot product. Cosine similarity in single-cell analysis measures how similar two cells' expression profiles are. Projecting a cell onto a principal component is a dot product, how much of the cell's variance aligns with that axis. 3 vectors in 3 dimensions. If the third is a combination of the first two, it adds no new information. The rank of a matrix is the number of truly independent directions. A 20,000 gene expression matrix rarely has rank 20,000. Gene co-regulation means thousands of genes move together. The true rank might be 50 or 100, the number of independent biological programs. This is why PISA works. The data lives in a low-rank subspace. Vectors, matrices, transformations, dot products rank. The drafting tools are laid out. Next, the special directions where the building resonates.